We're talking about the splicing of the pre-mRNA. For forming the mature mRNA, we have to splice the pre-mRNA that we just discussed. Pre-mRNA is formed after the DNA is transcribed, and then the pre-mRNA is then transformed into mature mRNA so that it can be translated. So what happens is that splicing of the pre-mRNA is the part of the process by which the precursor RNA is pre-mRNA is transformed into a mature mRNA. Alterations in the snRNP that we're going to discuss later. What is snRNP? Alteration in the snRNP assembly can lead to clinical disease. That is, it can lead to spinal muscular atrophy. And what happens is that there's snRNP assembly is basically affected due to decrease in SMN protein, and it leads to congenital degeneration of anterior horns or anterior motor cells, anterior horn of the spinal cord, symmetric weakness, and leads to symmetric weakness like floppy baby and floppy baby syndrome or hypotonia. So let's start the process of splicing a pre-mRNA. What happens is that for splicing the pre-mRNA, first thing that happens is that the primary transcript combines the primary transcript, that is the heterogeneous nuclear RNA, combines with the small nuclear ribonuclear protein snRNP and other proteins to form spliceosome. So that's what's written in our book. But what actually happens is that first the U1, that is U1 snRNP, binds to the 5' splice site, and this is where the intron exon junction, the first intron exon junction, and the U2 snRNP binds to the A branch point. That A is adenine. So basically what happens is that in the introns we have the donor site has GU sequence, right? That is give U, and the acceptor site has AG sequence, that is a gift, and between these two sites is a branch point, that is this A. This is basically a branch point in the middle which consists of adenine residue. And so the U1 snRNP first binds to the intron exon junction, the first intron exon junction at the 5' splice site, and the U2 snRNP binds to the A branch point. Next, what happens in the next step, there is cleavage at the 5' end. So there is cleavage at the 5', that is the donor site end, and what happens is that the phosphate which was attached to GU now binds to A, that is the A branch point. And basically this creates this kind of a loop, that is the lariat shaped loop, which brings the two exons closer together so that the OH at the exon 1 can attack the P near the exon 2. So this is the OH at the exon 1 that can attack the P at exon 2. The next step is cleavage at the 3' end at splice site, and the lariat is released to precisely remove the introns and join the two exons. So what happens in here is that the OH that was attacking the phosphate, what it does is that it replaces it. The OH gets attached to the AG, and the phosphate remains in the middle of the two exons, and now the intron is all gone. We have the two exons that is required to form the mature mRNA, and this is how the snRNP splice out the introns. If you form antibodies against snRNP, autoimmune disease leads to autoimmune disease. Anti-Smith antibodies are found in lupus. Anti-U1 RNP antibodies are also directed against snRNP and is found in mixed connective tissue disorder. Sometimes we export mRNA into the cytosol and do not need to use it immediately, so we can store it in the P bodies and processing bodies, that is the cytoplasmic processing bodies, P bodies, which basically stores it, and either it degrades it or uses it for future translation.